Hi, good morning. Well, I think they gave me the short straw here because I do have to use the word genomics. Dr. Lee kind of set me up for that, but let me see if I can make this work a little bit better for me since I'm short. So what do you mean? What do we mean by genomics? And the easiest way to think of this is it's all things DNA. So what is DNA? All right, well, what we're looking at here, and let me see if I can figure this out. Okay, great. This is a chromosome. And chromosomes are basically, I think it might be better. Yeah.
Solid.
Okay, thank you. So chromosomes are basically, one way to think of them is sort of like a big ball of yarn. And inside that yarn, what is that yarn made out of? Well, it's made out of these basic 4 things that are called base pairs. And they are actually, they have a sugar and they have a phosphate, and they're arranged in this kind of ladder that then encodes for various genes based on these combinations. And then it's all wrapped up in this what's called a double helix. And then it's folded up and surrounded by these proteins called histones into this very nice, neat little package, a chromosome. Now, one of the things, to say another dirty word that I think is going to be said this afternoon, epigenetic. Okay, what that refers to, if you start messing around with this kind of structure and you start looking or modifying some of these proteins or the way the proteins that this stuff is encoding for, that's considered more epigenetic. Now, everybody here knows chromosomes, so that we've got 23 pairs of them in every cell, and you have 22 that are called somatic, and then you have XX and XY. By convention, and this is actually a French dude who did this, the top portion of the chromosome is referred to as the P portion, and the bottom is the Q portion. So if you're starting to see where I'm going here in the next slide, hopefully that'll make some sense. So we've known for a long time that you can use chromosome analysis to study various blood cancers. And this has been done going back into the late 50s into the 60s for leukemias. But for many years, people said, myeloma, well, that's like not growing. We can't study those chromosomes because in order to do this, you have to have cells that are dividing. Normally, those chromosomes are not sort of packaged up in that ball of yarn. They're kind of floating around. So it wasn't until really, and this is a slide actually from the late 1980s from a French group, that they showed that in some patients they could do this cytogenetic analysis, and this is in myeloma cells, so this is not in normal cells, that they could start showing, hey, in some people's myeloma cells, we're seeing weird stuff. So we're seeing that here's chromosome 1. Well, it should only have 2 copies, right? So it's got these like weird recombination, as we call it. Here's a patient down here who looks like they have some switching of chromosome 11 and 14, which we would call translocation 11-14. So this, we started to recognize around the year 2000 that you could do this in myeloma in big ways. But then what happened is a technique is developed over here called FISH, which many people, everybody heard of FISH? Anybody heard of FISH? So what that stands for is fluorescent in situ hybridization. And what that turns out to be is a much, much more powerful tool to analyze myeloma because you don't need to do this. You don't need to have cells that are dividing. You can just take myeloma cells. Now, this is done on bone marrow, but you take those cells out and then you start sprinkling them with these various fluorescent probes and look at them under a special microscope. And then you can start finding out, again, about pieces that are being switched among these chromosomes. So, and the way they do this, for example, very simply, if we're looking for a translocation of the 14th and 4th chromosome, in other words, switching pieces, you can use these probes, one's red, one's green. If you see a red and a green probe there together, you know that that translocation is there. And then this one is showing you a deletion of P53 or deletion of a portion of 17. This is a very important tumor suppressor gene, so if it's missing, it means that the myeloma is kind of unleashed. But you can tell in this example, so you don't have the pair green and red, all of a sudden you're missing a red, and that goes along with the deletion. And this is how we do this. Now, this test is great. It works better if, when it's run on your bone marrow, that they sort out the plasma cells. That helps increase the sensitivity. But we use this now all the time. And we're starting to learn more and more things about chromosomes in myeloma. And I think we're only at the tip of the iceberg, really. This is a picture of chromosome 1. Now, you can see this is, I mean, it looks like a gobbledygook, but these are actually called bands. And it's a way we kind of try to organize the genes that are on these chromosomes. And we're now realizing that chromosome 1 abnormalities in myeloma, which really weren't thought about maybe 15 years ago, this chromosome is actually quite important in myeloma propagation. And not only are there pieces that get duplicated in the bottom Q part, but now we're finding out very recently that a missing portion up here in 1P. Now, these bands are numbered, so you can sort of map to where you have a chunk missing. But in this case, now we're learning right up here, if you're taking out a chunk, that's actually really bad. That's another cancer suppressing gene that we've just recently found out. If you're missing that in your myeloma cells, that's probably not a good thing. Okay, so this is one of the things that we use to define what is high risk in multiple myeloma. And this is a kind of a topic where people have various ideas about it, but we have some things that we think can tell you by high risk, meaning is your myeloma easier or harder to treat. So we just talked about some of these things with chromosomes here, the gains of what we call that 1q, the bottom portion of 1, a deletion. Now, why is chromosome 14 here all the time? That's not an accident. So on chromosome 14 is what's encoded the part of what we call the heavy chain of the antibody. That means the part that tells you it's IgG or it's IgA, not the light chain part of it, but this is what's called the heavy chain part. So that's why that's involved all the time. There are ways to analyze all of the genes simultaneously. This is something called a gene expression profile. There's a commercial one available called Sky 92 where they basically take the DNA out of the myeloma cells and squish it all up, and then they look for certain changes and do it in sort of a one-step method. And this is something that starting to be used, but most people here, I suspect, have not had that run on them. We're also starting to understand if you have not just one of these changes here, but if you have a couple of them, 2 or more, that seems to be something that means that your myeloma is going to be a little bit harder to treat. Now, there's some other things. This came out very tiny. I'm not sure why. But if you have some kidney problems, that can increase your risk, we think, over time. Now, more and more, we're looking at people who have plasma cells that might be in the blood itself, that you can check for that. That can be something that might be associated with a higher risk. If you have myeloma growing outside of bones, what we call extramedullary, that seems to be a concern. And finally, we have the staging system called the Revised International Staging System, or RISS, and we're going to take a look at that now. Okay, on the left, this is our staging system that was devised in 2015. It looks kind of complicated, but it's only looks at a couple of things. One is, do you have any of those cytogenetic risks? Yes or no. Do you have a high lactate dehydrogenase, or LDH? We're going to talk about what that is. Do you have a high beta-2 microglobulin, which is essentially a protein that you can measure that's associated with how much myeloma do you have? And we can take, and then do you have a normal amount of a protein called albumin, which is kind of a measure of how your nutrition is, how healthy you are. So you can take all of those and come up with 3 different grades, one of which is the top one called Revised ISS-3. Now, we know that people who fall into that category do have a higher likelihood of having harder to treat myeloma, but more recently, and this is what's shown here on the right, we're even revising this further because this old schema didn't include changes in that chromosome number 1, which we think are important. And this gradation now has 4 categories. And the hope here is that we can single out people who need more treatment, but also stop pulling back on treatment for people who fall in the best risk categories. So I think you're going to probably hear your providers talk more about this. Now, what is lactate dehydrogenase? It's an enzyme that all cells have, and it's basically something that's involved in cell metabolism and cell turnover. So if you have a lot of cells growing, you have a lot of LDH. And we've known again for a while that in lymphomas and leukemias, it's an important factor to check. Is it high or normal? But the interesting thing about LDH, it wasn't really adopted as a risk factor for myeloma until 2015, but there are reports almost 50 years ago, that this was an important thing to check. So anyways, just something that you should have checked at diagnosis. It does fluctuate, this particular lab, during treatment, so it's not so much important later on, but at diagnosis, this is something that we want to make sure that people get checked. Okay, now these categories that I mentioned don't cover all the risk that we know. So there are some what we call functional high-risk people with myeloma. That is, for example, a person who went through some of the treatments that Dr. Lee talked about, but let's say 6 months later, they've had chemo, they've had an autologous stem cell transplant, they're on maintenance, now their myeloma's back again. That's a person who essentially does have high-risk myeloma. We're starting to think about people who, if they acquire a new mutation, does that in their bone marrow cells, does that make them high risk? That may be true. And finally, if later on you start having myeloma growing outside of the bones, that is probably also a risk factor as well. Okay, so they also asked me to talk a little bit about minimal residual disease testing. So what we're trying to do again is to come up with ways to say, is your myeloma in a deep, deep, deep remission, more than just, okay, those protein levels are gone. And this is an area of a lot of research that's been happening really for the past 10 years and we're not ready yet with an answer. But basically, you're looking for that needle in the haystack. You're looking for that one myeloma cell that's that dot that Dr. Lee showed you that's still sticking around. Can we make sure that that's gone, or at least to the best of our ability gone? So this is, unfortunately, I'm sorry, it's not big enough to read. There's two ways that we do MRD testing, and this is usually done at the conclusion of treatment. One is by flow cytometry, and I'm just going to go right to this, which is shown here on the left. So what you do here is you take bone marrow, you pass it through a laser, the myeloma cells scatter light, and you can also mix those cells in with fluorescent tags again and sort of figure out which cells are in that bone marrow. And in this particular example, we're able to show that these red cells are actually abnormal plasma cells that are in this bone marrow. Nice thing about flow cytometry, it's relatively inexpensive. It's about $500 per test, which is in this healthcare system is not so bad. You also don't need to know what kind of myeloma the patient had at the beginning. You don't have to know that information. You can do it essentially at any time. The downside of it is if a person has very few myeloma cells in their bone marrow, the ability to pick up that one abnormal one is kind of low. So the sensitivity, as we call that test, can be kind of not as good as, say, some other tests. The other test that's out there is what's called next generation sequencing. And you may know this technique under the trade name clonoSEQ, if anybody's had that run on their bone marrow. But this is a way to use sort of a DNA-based test to look for basically certain DNA patterns that are in this sort of stew that gets created to look to see if you have any residual myeloma cells. Now, in order to say that you've got abnormal myeloma cells, you had to have had a sample taken essentially at baseline or at least at a time point when you had a lot of myeloma cells in your bone marrow. Otherwise, it doesn't work very well. A little bit more expensive, but what they can do is sort of track things longitudinally and give you a graph and say, hey, you had a lot of myeloma up here, but now we're testing later on and you have very little. Okay, so should we use this kind of testing? People ask that as a way to either quit chemo. Okay, my MRD is negative, can I just stop? Or if it's positive, do I need a whole lot more chemotherapy? And I think, I hope I will share the opinion of other people speaking today. I think we're not there yet. I think we have a lot of research still out there to help us decide that. Now, we do know that, and this is what this is supposed to represent, these blocks are, and names are a bunch of different myeloma studies. And what this doctor did, Dr. Munshi, is he put all this data together and he said, okay, if after treatment, and these are a bunch of different treatments, if you're MRD negative, do you do better than if you're MRD positive? So you're negative here in the blue, you're positive in the red. Okay, looks like it's better to be negative. But what this kind of result doesn't tell you is that maybe your myeloma was easier to treat, and that's how you got MRD negative. Kind of going back to what Dr. Lee was talking about, you didn't have those really tough clones emerging, so you did good. It wasn't that the test was so great, you just had better myeloma to begin with. And then this is a study that I was involved in called the MASTER trial, and what I'm trying to show you here, these are looking at cytogenetic risks 0, 1, 2. Okay, all of our patients in this study got to MRD negativity after periods of time, so that's great. We got a lot of people in very, very deep remissions, but what we ended up finding out is even though we had sequential MRD negative tests, people who had more cytogenetics, 2 or more of those changes, unfortunately still ended up having their myeloma come back much more than these people. So I think we do know that MRD negativity, even sequential ones, is still not the answer yet for what we need. And then Dr. Lee mentioned maintenance. I'm just going to not try to take too much time here. This was a study that was just presented a few months ago, trying to say, if we look at a person who after a stem cell transplant is asked to take lenalidomide, Revlimid, or not, can we actually analyze people and say, can they stop, after 1 year, 2 year, 3 years, 4 years? Now their analysis, and I'm not going to go into the details of it, but basically it is an analysis based on who's still around. So there's some issue with that kind of analysis, but suffice it to say, the conclusion that a lot of people took away from this talk was after 5 years of lenalidomide maintenance, doesn't look like you're doing a whole lot better than people who stopped it. And then if you are MRD negative, it also looked like maybe you're not getting as much lenalidomide benefit. However, if you are positive, and this is one thing where this may actually help us say treatment is better, still look like if you're MRD positive, you're doing much better, much, much better if you're staying on lenalidomide. And then finally, if people had more than one MRD negative test, if they had a couple, they're doing better too. So this may give us a hint that there are subgroups of patients that can stop maintenance at some point. Okay, finally, I just want to touch on scanning in terms of ways to also determine risk, but also treatment cessation. This is on the left here is a study from Mayo. And this is a much larger randomized trial from France. What they did here is they had patients go through treatment, and in this case, a transplant. And then they said, okay, we're going to do a PET scan. How many people here have had a PET scan? Okay, a lot. Okay, so what they did is they said, after the end of your treatment, we're going to do a PET scan. And we're going to see if it's negative or positive. If it was negative in the red here and the blue here, you actually did a lot better. So that is another tool out there that may be used in the future to say, okay, if your PET scan's still positive, you need more treatment. So in summary, I think we're beginning to know a little bit more about risk than we did before. We still can't identify everybody who has high-risk myeloma. And I'm sure that there is somebody either that you know or in the audience who may have been told at the beginning of their treatment, hey, this doesn't look so difficult, but maybe now that you're on your 6th or 7th line of treatment, that's not the case. So we really do understand. I think we all who treat myeloma patients have a lot of humility about this, much, much more to learn, but we're really happy that we have some of these tools here now. And I think in the next few years, we really are going to be looking for those two twin goals: less chemotherapy for people who don't need it, but more chemotherapy for people who do. So I will stop there. Thank you.